Okay, so we talked about how genes are transferred. Let's talk about some examples of those genetically transferred, those genetically modified organisms. So IB wants you to know two named examples. So when you see this question, make sure you include the name and give as much detail as you can. But we already talked about one. We talked about transferring the human gene of insulin into a bacterium. So that's a genetically modified bacteria. But let's talk about a couple more just to play it safe. So there is a type of bacteria. We'll do it like this. Because it's called Bacillus thuringiensis, but we'll call it BT, and it makes a type of protein, which I'm just going to squiggle like this. If it gets inside of an insect, it kills the insect. So here's my dead insect. It's upside down, legs up, crosses for eyes. So what happens is this protein interferes with its digestive system, and it ends up dying. But somebody had the great idea of saying, what if somehow we can have plants produce this protein, and then we wouldn't have to spray plants with pesticides and things like that? So they did that. So what they did was they put the gene inside corn. It's my corn plant. So they took the gene out. So let's make the gene red for our sake. So here's the DNA and the protein, and they ended up genetically modifying plants to have that DNA inside. So the plant was automatically making this protein. So when insects tried to eat the plant, they died. And this plant, this corn, has become quite popular, and it's called BT corn. BT corn. We're going to talk about the pros and cons later on. I mean, there's definitely a lot of pros you can think of right away, like, hey, we don't have to spray poison on this plant, which is not good for humans. But there's also some bad things. So transfer the gene from a bacteria into corn. Corn starts producing this protein that kills the bugs. A second one is how we genetically modified mice to contract polio. So mice, and why would we want to do that? Mice do not have, so if we look at a mouse under a microscope, on their cells, they do not have that specific protein that invites the virus in. Humans do. So what they did was they took the gene for that specific protein out of humans. They put it into the mouse genome, and now the mice started producing this protein on the outside of their cells. So when the polio virus comes, and that's how viruses work, it attaches to the surface protein. The virus ends up inside the cell, and this is what happens when you get a cold or the flu, or it's how coronavirus works, and also HIV. The mouse can get polio. So why would we want mice to get polio? So the reason they did this was so we could test certain vaccines on mice, right? That's supposed to be a needle. Certain vaccines on mice to see if it makes them immune to polio. My thing just froze. So those are two examples. So it's BT corn and also mice that can contract polio. My pen is going nuts. That's back to normal. All right, thanks.